Okay, so it's time to start. Thank you everybody for coming to another edition of the Virginia Tech Life Science Seminar Series. It is my pleasure to be your host today and introduce our guest, Professor Steve Mansoorabadi. Steve obtained his undergraduate degree at the University of Wisconsin in Milwaukee. And there he clearly showed his interest in science and his diverse interest in science. He had 4 different majors: mathematics and physics, chemistry, biology, and also biotechnology. From there, he moved to University of Wisconsin in Madison to pursue a PhD in biochemistry at the world-renowned Enzyme Institute under the tutelage of Professor George Reed. And then there, he actually became an expert on radical chemistry. And one of the main techniques that he used was EPR. And just want to mention that being at the Enzyme Institute was a real plus, because he had the opportunity to have as part of his committee people that you might know, like the late Mo Cleland, Perry Fry, Tom Record, and George Phillips. So all providing crystallographic expertise, physical chemistry, kinetics, biochemistry, etc. He was quite prolific as in his PhD. He published, I think, more than 7 papers, and he also was an NIH fellow. From there, he moved to University of Texas in Austin to work with Ben Liu. He also he obtained a postdoctoral fellowship from NSF, from NIH. And he also was quite prolific, including maybe more than 10 papers, some of them in JACS and also Nature. Again, focusing on mechanistic enzymology, looking at mainly at radical chemistry. And then in 2012, he started his independent career at the Auburn University in the Department of Chemistry and Biochemistry. And he has been also quite prolific. He has published many papers, some of them in quite high-profile journals such as Science. And he also has been very good in obtaining funding in a very difficult environment. He has a career award from NSF. He also has a career award from the Department of Energy. And he's a co-PI also in other DOE and NSF grants. And throughout the years, he has been developing techniques and studying biosynthesis and methanogens and also bioluminescence. And that's what he's going to summarize today. Thank you, Steve. And I need to press this here too. This is for you.
Thank you. All right, thanks, Pablo. Very kind introduction. Thank you also for the invitation to come here. So I'd like to talk a little bit about my work. I started in 2012 at Auburn University, and so my lab was sort of focused on the tetrapyrroles as a class of molecules. So we're interested in sort of the chemistry and biochemistry of tetrapyrroles, how they're made, how they're used, and various applications of them. So this class of molecules is referred to as the pigments of life. So they're pigments because they're colored. They range from the red of heme, the green of chlorophyll, yellow for this Coenzyme F430. So they have various metals. We have iron, cobalt, magnesium, nickel can be chelated in the center of these molecules. And they're also the pigments of life in that they're found in all domains of life, from archaea to bacteria to eukaryotes. And they're very important, and they're found, they play key roles in many very important biological processes, from aerobic respiration, denitrification, amino acid metabolism, and so on, photosynthesis. So they're very widespread, and they have many applications. So we have projects that are sort of related to all of these molecules shown on the board, but today I just wanted to focus on 2 of our projects related to bioluminescence and methanogenesis, so related to dinoflagellate luciferin and Coenzyme F430. So I'll begin talking about Coenzyme F430. So it's one of the key cofactors in methanogenesis, so it's found in methanogens. I'm sure you guys are well aware of methanogen biochemistry here. Methanogenesis, as the name implies, is the formation of methane. And methane is the major component of natural gas. Natural gas accounts for nearly a quarter of the world's energy demands. And very recently, we've discovering all these shale formations, and so the production of natural gas from these unconventional sources is almost increased exponentially in recent years. And it's estimated that the reserves of natural gas could maybe supply enough energy to sustain us for the next 100 years or so. But unfortunately, natural gas is often found with petroleum. So when companies are extracting petroleum, they don't really care about the natural gas. So they either flare it or vent it into the atmosphere, which is very wasteful and impactful on the environment. Actually, this is a heat map taken from a satellite, and you can see all the different places around the world that flare methane. So that's pretty frightening, I think, when you see it on a global scale, the production of these very potent greenhouse gases. Actually, methane, as we mentioned, is a major component of natural gas, and it has 30 times the global warming potential of carbon dioxide. So venting it is probably a bad idea. But methane is also produced, as we mentioned, naturally by these methanogenic archaea, and it's estimated they produce nearly 1 billion tons per year of methane. But a lot of that is captured by methanotrophs that eat the methane, and so it's not actually released up into the atmosphere. So shown here is one strain of methanogens which we work with in the laboratory, this Methanogenesis acidivorans. And it sort of has a dubious honors. It was sort of linked potentially in a fairly recent PNAS paper to one of the largest extinctions ever. So in this paper, they sort of tried to estimate at what point during evolution did this strain acquire the ability to produce methane from acetate in the oceans, and they put it right around the time of this mass extinction. So it's sort of speculation that this contributed to one of the major extinctions where nearly 90% of all species died. So it kind of caused a runaway greenhouse effect that was partially responsible, potentially. So that kind of, I think, is a cautionary tale that we should be careful on how much greenhouse gases we're emitting in the atmosphere. But anyway, so how do these methanogens produce methane? Well, they use this enzyme, methyl-coenzyme M reductase, and it uses at its active site this nickel-containing tetrapyrrole, Coenzyme F430. And so the reaction is shown here. It takes methyl-coenzyme M and coenzyme B and produces this heterodisulfide, CoM-CoB disulfide, and releases methane. So it uses this nickel catalyst to carry out that reaction. It needs to be in the 1+ oxidation state for it to be active. And recently, a homolog of MCR has been found in methanotrophs, these anaerobic methanotrophic archaea, or ANME. And they actually use this enzyme to run the pathway backwards, sort of reverse methanogenesis. So it's thought that MCR will catalyze the oxidation of methane using this enzyme and this catalyst. So there's a lot of interest in maybe using these methanotrophs to maybe create a fermentation process and put it at these sites. So instead of venting or flaring methane, we can just feed it into a bioreactor and convert it to something useful. So there's a lot of interest in maybe using these methanotrophs or designing new ones that can be used for that purpose. The problem is you can't really use these ANMEs directly. They grow very slowly. I forget the doubling time is like once a month or something, so it's like super slow. And plus nobody can currently get them in pure culture. So it's very difficult and not really amendable for use in a process like this. So ideally you'd like to be able to engineer a strain that can use MCR for this purpose. But before I started, there really wasn't much information about how do you make F430. So if you can't make the cofactor, you can't make the enzyme active in a heterologous host. So that's kind of what we focused on. We were interested in trying to figure out how to biosynthesize this cofactor. So if you can do that, then you can maybe develop an engineered strain of methanogens using this enzyme. Also, if you know how to produce F430 and then therefore also produce active MCR, you can also maybe inhibit that pathway specifically. So you can use that to maybe try and make an inhibitor to reduce natural greenhouse gas emissions. So that there's actually a lot of interest in feedstock. So one of the major producers of methane are livestock, right? The archaea in their guts will release some of their feed as methane into the atmosphere. So if you could include an additive in their feed that inhibits this process, maybe you can reduce some of those emissions. And then maybe some of that carbon will get diverted to making the cows fatter, and you can get more money out of it. So there's some interest there as well. How do we get started in this? So we sort of went back to the literature and tried to figure out what is known about tetrapyrrole biosynthesis in general. And so there's two main pathways. You can either start from glutamate attached to a tRNA or from glycine. And these two pathways converge at this 5-amino livulinic acid. And then 8 of these are stitched together to form uroporphyrogen 3. And this is the last common precursor of all tetrapyrroles. So all tetrapyrroles have uroporphyrogen 3 as an intermediate. So you can either decarboxylate this molecule to form coproporphyrogen, and you can make things like heme and chlorophyll, or you can methylate it to form the methylated tetrapyrroles like cyroheme, B12, and also coenzyme F430. So we figured probably precorin 2 is a good guess for a starting point in the biosynthesis of F430. So what do we have to do to convert precorin 2 to F430? Well, there's a number of things that have to happen. Most obvious, you have to insert nickel into the center of the molecule. There's also 2 of these carboxylic acids have to be converted to amides. And then, somewhat less obvious, you have to reduce the ring by 4 electrons to get to the same oxidation state. And then you have to cyclize here and here to make F430. So we figured we needed enzymes capable of doing all those reactions, right? So we just sort of did some comparative genomics. We were gazing at genes and trying to see if we could find a cluster of genes that seemed like it encoded enzymes capable of that chemistry. And we were lucky enough to find one that we thought was a pretty good candidate. So it was a cluster of 5 genes, which we called CFB A, B, C, D, and E. So these are conserved. They're found in all methanogens. So every methanogen has a homolog of each of these genes. Interestingly, though, it's only these genes were only clustered in 1/3 of the genomes that I looked at. They were partially clustered in 1/3 and then completely scattered in another 1/3. So if you're unfortunate enough that, I guess, came in at the right time where there was enough genomic information to have some that were clustered. Otherwise, we might never have found them, right? So that's kind of shows the importance of these, massive, data can really help in identifying clusters. So what are each of these genes homologous to? Well, the first one, CFBA, was homologous to the cyrohydrochlorine cobaltochelatase. So this is an enzyme found in B12 biosynthesis. It catalyzes the insertion of cobalt into cyrohydrochlorine. So it was a metal ion chelatase, which inserts the metal. And obviously, we have a metal that needs to be inserted, so that made sense that there should be a chelatase homolog in the pathway. Then the other gene, the CFBB gene, it was homologous to this coborinic acid AC diamide synthetase. It's also involved in B12 biosynthesis, and it catalyzes the amidation of the A and the C carboxylates of coborinic acid, and it's the exact same positions in F430, so that made sense. We should have a homolog of this gene in our cluster. Let's see. Okay. The next one, there are 2 actually that work together, and they were homologous to nitrogenase, which at the outset doesn't look very relevant. Nitrogenase is this 2-component metalloenzyme, which is involved in nitrogen fixation. So it reduces dinitrogen to ammonia. So it's basically an 8-electron reduction, 6 electrons to reduce nitrogen to ammonia, and another 2 to reduce 2 protons to H2. So this reaction, it's a very energetically demanding reaction, requires 16 ATPs to carry out this reduction. So I mentioned that you have to reduce the F430, the precorin 2, by 4 electrons in order to get to the redox level of F430. So maybe it could be playing a role in that reduction process. And sort of supportive of that, there is another homolog of nitrogenase involved in chlorophyll biosynthesis, this dark or protochlorophyllide oxidoreductase. So this enzyme catalyzes a reduction of a single double bond in the biosynthesis of chlorophyll. So there's some precedent for this scaffold of an enzyme to be able to accept tetrapyrroles and reduce them. So that made a lot of sense. The last gene in the cluster, CFBE, is homologous to this MUR ligase. So MUR ligases are involved in peptidoglycan biosynthesis, which is kind of odd. It doesn't seem like it really fits. So basically it forms a peptide bond. So it activates this carboxylic acid with ATP to facilitate a CN bond, a peptide bond, to make this intermediate in peptidoglycan biosynthesis. But if you're going to form a ring in F430, specifically this carbocyclic ring, you have to activate a carboxylic acid and form, in this case, a CC bond. So you can maybe use similar chemistry to carry out that transformation. Or at least that's what our thinking was. So this 15-17-3 seco F430 17-3 acid, I didn't name it. So this is actually an intermediate which was found in methanogens, which suggests that the last step in the pathway is formation of this carbocyclic ring. So that's a key point. So we went ahead, kind of, I don't know, naively or bravely, as a young faculty member, we went through and cloned all of these genes and set about testing their activity for these reactions. So we cloned them all as his-tag fusion proteins in E. coli. We're hopeful that it would work out. And then the next thing we had to do is prepare the substrate. So you can't just buy precorn 2 from Sigma or something. So we had to enzymatically synthesize it. So you can buy porphobilinogen. And then we used the heme C and heme D enzymes from E. coli. We just cloned them from there to make this last common precursor of all tetrapyrroles, uroporphyrinogen 3. And then we cloned this SIR-A, the methyltransferase from Methanosarsina, and synthesized it in vitro. So we were able to obtain our substrate for this potential chelatase. So then we set about trying to assay it. So we mixed our synthesized substrate with our his-tagged CFBA chelatase enzyme and added nickel chloride as a source of nickel. But we didn't see any activity. Nothing happened. So that was a little discouraging. I thought, well, maybe this his tag is interfering with the activity of the enzyme. So we chopped off the his tag using thrombin and then added in the nickel. And we didn't see any activity. So that was pretty disappointing. But we went back to this. The homolog is actually a cyrohydrochlorine cobaltochelatase, not a precorn 2 cobaltochelatase. So to prepare cyrohydrochlorine, there's another enzyme, the SIR-C, which is a dehydrogenase. So it uses NAD to oxidize precorn 2 to cyrohydrochlorine. So cyrohydrochlorine is the actual substrate for the cobaltochelatase. So we thought, well, maybe we have to go an extra step. Let's oxidize it to cyrohydrochlorine and see if now that will be a substrate. So we took our precorn 2, we incubated that with SIR-C and NAD. Also, this was purified from Methanosarsina, or cloned from Methanosarsina, purified in E. coli. And the precorn 2 solution, which is kind of a yellowish color, turns this very pretty magenta. And then we can run this on the HPLC. And this compound has a retention time around 17 minutes. It has a very characteristic UV-Vis spectrum of cyrohydrochlorine. And the mass is also consistent with cyrohydrochlorine, so we were able to, sorry. Yeah, right, yeah. Sorry, we were here. So we were able to, maybe I'll just do it one by one. We were able to successfully synthesize cyrohydrochlorine. So then we took that cyrohydrochlorine, we added our CFBA again with nickel, and now the solution turns from magenta to this almost a wine color. So it seemed like something was happening. We ran that on the HPLC, and then we see a new peak. So the 17-minute peak disappears. We get a new peak around 21 minutes. And it has a very different UV-Vis spectrum. It looks very similar to other metallated tetrapyrroles, so we're pretty excited that maybe nickel was actually getting inserted. And then we confirmed by mass spec it has the exact mass of what you would expect for nickel cyrohydrochlorine. So we confirmed that, yes, this enzyme is active, and it had to be cleaved. The his tag had to be cleaved. With the his tag, it wasn't active. So that is capable of catalyzing nickel insertion into cyrohydrochlorine. So then we took this enzyme, we added the next one in the pathway, and so which we think was the amidation enzyme. And so you need to include glutamine as a source of ammonia for the reaction, and ATP to activate each of the carboxylates for the substitution. And so when we did that, the color didn't really change at all. It looked exactly the same. However, when we look at it in the HPLC, the retention time shifts back to around 17 minutes or so. The UV-Vis looks identical, which is consistent with it not changing colors, but the mass is 2 units less than the nickel cyrohydrochlorine. So that's consistent with converting two carboxylic acids to amides. So it seemed like this enzyme was active. It could accept nickel cyrohydrochlorine as a substrate and convert the A and C carboxylates to amides. So that was good. And now is sort of the difficult step. Can we purify these metalloenzymes, a homolog of nitrogenase from E. coli, and get it to reduce this molecule? So we incubated this product of this reaction with this nitrogenase homolog. We included ATP, as is required in the nitrogenase reaction, and then we used dithionite as a reductant. And the solution turned yellow, which was very exciting because also F430 is yellow. So we're getting closer to F430, right? And then there's a dramatic shift in the HPLC. The product, we get a new product around retention time around 8 minutes. And it had this UV-Vis spectrum with absorption close to around 430. And its mass was actually consistent with the only intermediate that's known, this SECO F430 in the literature. And actually the UV-Vis spectrum of this compound is identical to this one. So we were pretty excited that actually this enzyme not only does the reduction, but also facilitates the cyclization of the lactam ring here. Then the last thing, we included this SECO F430. We added our ligase and included more ATP. And it turns from yellow to yellow, so not much of a color change. There's a very tiny shift in the HPLC retention time, but the UV-Vis changes pretty dramatically, and it's a dead ringer for F430. And the mass, again, is consistent with F430. So we're pretty confident that this was actually the cluster involved in making F430. Just as further identification, we took actual MCR from a methanogen and extracted the F430 and ran it on the HPLC. And everything is identical, the retention time, the UV-Vis, and then also the mass spec and the isotope distribution is exactly like F430. So, so we were able to find this F430 cluster, and the overall pathway is shown here. So the first step, we don't actually use precorn 2 as the immediate precursor. We first have to oxidize it to cyrohydrochlorine. Then we can insert nickel and then convert 2 of the amides to make nickel cyrohydrochlorine AC diamide. And then we use this nitrogenase homolog. So instead of reducing by 4 electrons, we have to reduce by 6 to get to the correct oxidation state. And then we can cyclize to make F430. So that was great. But our goal ultimately was to be able to produce this MCR in a heterologous host. So we're trying to work towards that. So the first step, we'd like to be able to recapitulate this biosynthetic pathway in E. coli or some other organism. So we went ahead and we cloned, we used all these duet vectors, and we cloned each of the F430 biosynthetic genes into these vectors. So we have CFB, A, B, C, D, E, and then the SIR-A and SIR-C enzymes, which are used to make cyrohydrochlorine. And then we co-transformed E. coli with all of these vectors, and then extracted out the tetrapyrroles. We added, supplemented it and tried to see if we could see F430 being produced in vivo. So during our extraction, we get a bunch of flavin, which is just endogenous to E. coli. We also see some shunt product to form this oxidized uroporphyrin. But the major peaks here, we have nickel cyrohydrochlorine, and we have nickel cyrohydrochlorine acediamide, which is the predominant product produced by this transformed E. coli strain, but we don't see any evidence for SECO F430 or from F430. So it seems to be stalling at the nitrogenase reaction, which is maybe not too surprising. So I think one of the problems with the reason it didn't work is you can't just feed dithionite to E. coli and get it to work, right? So you have to use an endogenous reductase that's present inside E. coli. And they don't really have, I believe, a ferredoxin that's sort of compatible with this enzyme. So regular nitrogenase uses either ferredoxin or flavodoxin is the electron donor during the reaction in vitro or in vivo. So we wanted to go ahead and try and find some sort of alternative physiological reductant to carry out this reaction. And methanogens, they do things kind of exotically. They have sort of complicated metalloenzymes that can use, say, hydrogen to reduce ferredoxin. And we didn't want to have such a complicated system. We'd rather use just a ferredoxin reductase, hopefully using NAD or NADPH as the reductant. And so we had some spinach ferredoxin and ferredoxin reductase lying around in the lab. So we tested it out. So spinach ferredoxin will take the small molecule protein ferredoxin, which has an iron sulfur cluster, and uses NADPH to reduce this ferredoxin, and then that can transfer the electrons to nitrogenase. So we used that, tested it out to see if it could reduce the reductase component of our nitrogenase homolog. So if you use dithionite and reduce this iron sulfur protein, you get a mixture of this high spin and low spin iron sulfur cluster. And then if you reduce it with the spinach ferredoxin, actually it reduces almost completely. But interestingly, the ratio of high spin to low spin is very different. It's almost exclusively in the low spin configuration. So we think it might actually be better. And our protein might be heterogeneous initially, and if you use dithionite, you get a mixture of species, but you get an exclusive low spin with this ferredoxin. So it seems like we're reducing our reductase, so it should be able to support catalysis, and indeed it does if we set up an in vitro assay using our CFB-CD enzymes and spinach ferredoxin. We're able to convert nickel cyrohydrochlorine acediamide to SECO F430. So this seems like it can support the reaction in vivo. And so what we're trying to do then now is include the spinach ferredoxin along with all of our other genes and growing those all in E. coli. And hopefully we're going to see some F430 being made in E. coli. One of the other things, the next step after we show in vivo F430 synthesis is getting the whole MCR assembled. And that's going to be what I spend the rest of my career on, probably. It's a very complicated system. So MCR, it's an alpha-2, beta-2, gamma-2 heterohexamer. And it has two molecules of F430 in the active sites. And there's several unusual post-translational modifications in the active site. So we have methylhistidine and methylcysteine, which is not too unusual. They are methylated amino acids, so you're methylating nucleophiles. So you can use a regular SAM-dependent methyltransferase for that type of chemistry. But we have two sort of unusual methylations. We have a 2-methylglutamine and a 5-methylarginine. So those are the methylation of unactivated CH bonds. So you're going to have to probably use radical chemistry to do that. And then there's a thioglycine. So the oxygen of the peptide backbone is replaced with sulfur. And the methylarginine and the thioglycine, this is the only enzyme that I'm aware of where these modifications are found. So it's very unique. So you have to figure out how to introduce all of these modifications and then insert F430 and get everything assembled together. It's going to be a pretty complicated process, but that's what's going to have to happen in order to engineer a methanotroph, right? So that's what we're working on currently. Oh no, what did I do? Sorry. So just to summarize this sort of first half of the talk, so we were able to identify the F430 biosynthetic gene cluster in methanogens, and we're able to produce all of these enzymes in an active form in a heterologous host from E. coli. So that's promising, working towards engineering a methanotroph. So this pathway involves the first nickel-specific chelatase. All the others that were known previously are either cobalt or iron or magnesium. So this one's specific for nickel. And it involved the formation of a previously unidentified tetrapyrrole, this nickel cyrohydrochlorine AC diamide. But really the key kind of magical step in the whole pathway is catalyzed by this homolog of nitrogenase. So it catalyzes a 6-electron reduction and a cyclization reaction all in one step, which is pretty remarkable. And I say it's a primitive homolog of nitrogenase, so it was noted previously in the literature that these methanogens had a homolog of nitrogenase. Sort of phylogenetic analysis suggests that really this enzyme evolved first. So I guess nitrogenase is a homolog of this enzyme, really. But so anyway, so that's pretty cool. That was our first project sort of related to methanogenesis. So I kind of want to shift gears and talk about the second project in the lab. This one is more of a computational project, or at least what we've done so far. And that's related to bioluminescence, specifically bioluminescence in dinoflagellates, which is pretty unique. So dinoflagellates, they're eukaryotic microorganisms. They're found both in freshwater and marine environments. And so several species, they're both photosynthetic and bioluminescent. So they do photosynthesis during the day and they bioluminesce at night. So it's regulated on a circadian rhythm. Okay. So here are two species of dinoflagellates that we work with in the lab, this Pyrocystis fusiformis and Pyrocystis lanula. So these are, these organisms are capable of bioluminescence. So what is bioluminescence in general? I'm sure you're aware it's the production of light by living organisms, and it can be used, actually it's been proposed to have evolved more than 30 times throughout the course of evolution, so it's very diverse in its uses in nature. So it can be used for communication and courtship in the case of fireflies. It can be used as predation in the case of this anglerfish, so it tracks the prey, in this case Dory and Marlin in Finding Nemo. It can also be used for camouflage. So this is a bobtail squid. It's kind of a cool, it uses counter illumination. So these guys are found in shallow ocean waters. So normally the sun would shine through and it would cast a shadow of the predator as it's swimming along. So they have a symbiotic relationship with bacteria on their belly, and it produces light, so it gets rid of their shadow, so they can hunt without a shadow, which is kind of slick. And dinoflagellates, they actually use it for defense. So it's sort of counterintuitive, but so their bioluminescence is induced by physical agitation. So if you shake them, they glow. So the idea is if a predator is chasing after a dinoflagellate, it'll disturb the water and they'll light up, and that's sort of a burglar alarm. And they'll alert the location of the predator of their predator to come and eat the predator. So it's a burglar alarm hypothesis. It's kind of a help me signal to the predator of their predator. Right? So that's kind of the fun part of dinoflagellates, but several are also capable of producing very potent toxins, such as saxitoxin or brevetoxin. So these are things responsible for shellfish poisoning. So they're very potent neurotoxins. They can cause diarrhea, amnesia, even death or paralysis. So they're very potent toxins. Dinoflagellates, they're also responsible for red tides. This is a harmful algal bloom. This was, I think, taken about the time I started at Auburn off the coast of Australia. So these dinoflagellates glow to very high cell densities, and then they can produce these toxins. So it has a very negative impact on the ecosystem, and also it's very costly from public health and also tourism industry. So it's estimated in the seafood industry, so almost over $80 million annually cost from these red tides. Also, unfortunately, it's the namesake of some other university in Alabama who hopefully will be worse at football this year. But anyway, so Crimson Tide, right? All right. So what's going on in these dinoflagellates? So they actually use a tetrapyrrole, this dinoflagellate luciferin. So it's an open chain tetrapyrrole, and it uses molecular oxygen and an enzyme called the luciferase. So this is the luciferin. So it uses molecular oxygen to oxidize this position to a ketone. So somewhere during that process, it generates an excited state, which relaxes down and emits light. So it emits light around 474 nanometers, sort of a bluish green color. But it's not really known the mechanism of this enzyme and sort of the nature of this excited state, which is relaxing down. So we were sort of interested in the mechanistic enzymology of this process. How does it work? So I mentioned that this enzyme, luciferase, so the bioluminescence is induced by physical agitation. So what's thought to happen, so this enzyme, this luciferase, the luciferin substrate, and also a luciferin binding protein are found within these subcellular organelles within the dinoflagellate called a scintillon. And it's thought when you shake a dinoflagellate, there's a receptor on the protein surface which can detect shear forces, and that leads to a signal transduction cascade, increase in intracellular calcium, and that depolarizes the membrane of these scintillons, opens up a voltage-gated ion channel, drops the pH, and turns on this enzyme. So this enzyme is regulated by pH. So it's completely off at pH 8, and it's maximally active at pH 6. Okay? So this protein itself is a large protein. It has 3 homologous catalytic domains. And so the structure of the third catalytic domain is shown here. So this is sort of a demonstration of the pH profile for this domain 3. So again, it's sort of almost completely inactive at pH 8 and maximally active at pH 6. And if you chop off the N-terminus of this domain, or if you mutate any of these conserved histidine residues to alanine, then you get an increase in that relative activity at pH 8. So it's thought that maybe these residues are involved in this pH sensing and the regulation of this enzyme. So we were interested in testing that, and so in this structure paper, this was done by Willard and Hastings at Harvard. They did a quickie MD simulation, 10 nanoseconds, where they made the quadruple alanine mutant of this enzyme, and they think that this lid is sort of blocking the active site. So this is a beta barrel, and you have this alpha helical lid, which is sort of near this N-terminus. And the thought was, so in what they observed in their simulations, if you convert all of these to alanine and run it for 10 nanoseconds, the barrel expands slightly. So maybe that's opening up access into the barrel so the substrate can go in and the enzyme can process it, right? So we wanted to test that maybe a little bit more rigorously and computationally. And also, it's not really clear if the alanine mutants, the observed structural changes, would be consistent with what's induced by pertonation, right? It's not quite the same thing. So we'd like to be able to run a simulation, MD simulation, look at the timescales. So we want to do it long enough so that we can observe a conformational change. So sometimes, their original simulation was only 10 nanoseconds. So that's not that long. Some conformational changes take much longer, maybe all the way to millisecond timescale. So we need to be able to either run it for a really long time or enhance the sampling so that we can observe the conformational change. And then we also want to be able to let the residues change their protonation state naturally in response to pH. So there's two sort of recent or somewhat recent innovations in molecular dynamics. So there's constant pH molecular dynamics. So you can set the pH as an external parameter and observe pertonation state changes during the simulation. And then you can also use accelerated molecular dynamics, so you can extend the timescales and allow you better sampling of conformational space. So we coupled these two together and tried to explore our enzyme and see if we can get some insight about how this enzyme turns on. So we first we looked at these histidine residues, the ones that were proposed to change their pertonation state and open up the enzyme. So at pH 8, so these are the 4 histidine residues, 899, 909, 924, and 930. So in the simulation at pH 8, they all remain in the deprotonated form. So that's consistent with the hypothesis. And then you switch to pH 6, and they all become protonated. So that's kind of consistent with what was originally hypothesized. But they have slightly different behaviors. So this histidine 909 is pretty much always protonated throughout the whole simulation at pH 6. This 924 kind of jumps back and forth and then increases. But these two here, there's kind of a lag. They don't get protonated right away. And then after 100 or 200 nanoseconds of accelerated molecular dynamics time, then the fraction of protonated increases. So it seemed like there was some sort of conformational transition occurring around this time, right? And we also noticed another pair of histidine residues, this 1064, 1065, so this histidine dyad, which is right on the bottom of the regulatory domain. It sort of stacks right on top of the barrel. And those also undergo sort of this delayed protonation during the simulation. So then we looked at this one of the histidines of that dyad, and we see that it's stacking right next to this arginine residue at pH 8. So in this closed conformation, in the crystal structure, the distance between the C alphas of these two is around 13 angstroms. But during our simulation, it actually gets even smaller. So it starts off around 10, and then there's this little conformational change. So this distance stays around 6 angstroms between the C alphas. So it's really tightly packed, even allowing it to move around, right, during the entire simulation at pH 8. And then when we look at what happens in pH 6, so we make it acidic, and it stays around 10 angstroms or so, and then it just expands dramatically right around the time when these residues become protonated, right?
Sorry, quick question. Mm-hmm. Which one is the physiological pH for this?
Right. Yeah. So in the sort of resting off form, it's around pH 8, and then it gets acidified to around 6. So exactly, the scintillons, they're pH-dependent flashing, so you can actually purify the scintillons and study them, and they behave exactly like the enzyme itself. And I think they also tested the physiological pH, and it expands in that range. Yeah. So right after around 300 nanoseconds at pH 6, there's a really dramatic conformational change. And so this is the distance between those two C alphas, and it sort of fluctuates between about 17 angstroms and almost 50 angstroms away from each other, right? So if we look at that residue, so here's the after 500, two different time points after it opened. So there's sort of the 17 angstrom distance between the histidine and that arginine. So this is sort of the closed form. It's still much more open than at pH 6. So this can be here or can flip all the way out and be about 50 angstroms. So it actually converts the alpha helical bundle, which was stacked on top very tightly, it tips over, and then this histidine dyad sort of converts into a dynamic loop and can go back and forth, right? So now it really opens up the active site so that substrate could potentially come in. And the arginine actually flips out from its position interacting with the histidine, and it forms a salt bridge with this glutamate on the exterior of the protein. Okay. So that was pretty interesting. One other residue that we noticed, which was kind of weird. So at pH 8, there's this lysine residue, which is on the surface of the protein over here. And it's exclusively found pretty much in the protonated form at pH 8. So at high pH, it's almost completely protonated. But then at pH 6, it actually becomes deprotonated. So it's opposite of what you would expect. And so we noticed here that we have this arginine residue. There's this lysine, the arginine. There's an arginine near the lysine residue. And then also we have the N-terminus, which is very dynamic at pH 8, and it has this glutamate residue. And at pH 6, the N-terminus reorganizes and forms a helical bundle, and it forms a tight hydrogen bonding interaction with that lysine and the arginine residue. So they all come together. So we see this reverse protonation effect occurring on the exterior protein, and we think it's linked to this conformational change that occurs. So it becomes buried and becomes deprotonated, right? And forms this hydrogen bonding interaction. Okay, so what about the active site? What's going on there? So there is a lysine residue, which is in the bottom of the barrel, near where the presumed active site is, and it's completely deprotonated at pH 8. And then when you shift it to pH 6, it becomes protonated. So as the active site becomes more solvent accessible, it becomes protonated. And then it sort of induces a change in the barrel. So if you kind of notice, here's the barrel at pH 8. At pH 6, it becomes much more expanded. And so there's two arginine residues which are kind of stacking above the lysine residue. So that kind of leads to an expansion and a reorganization of the active site. And then we have this glutamate residue, which is up here. It moves about 7 angstroms into the active site and positions next to the arginine residues. So the substrate of this dinoflagellate luciferin substrate, it has 2 carboxylic acids. So it's likely that these arginine residues interact with the substrate active site, and it's maybe positioning this glutamate to serve as a catalytic base. So as the pH decreases, it also reorganizes the active site and converting it into an active form. So that was something this is hot off the press, or actually pre-press. We just submitted this to biochemistry. We'll see how that goes. But I think it really shows kind of the power of this MD simulations and is to applying it both accelerated and constant pH to observe conformational dynamics of these pH-regulated proteins. And how am I doing on time? 12 more. Okay. So then the other computational aspect to this, so we wanted to sort of computationally investigate the pH regulation of this enzyme, but we were also interested in sort of the chemistry, what is the nature of this excited state. So instead of using MD for that purpose, we turned to density functional theory, ab initio quantum chemistry, to see if we could get some insight into what's going on in this reaction. So just as a reminder, so we have this luciferin substrate. It's an open chain tetrapyrrole. So this enzyme, once it becomes activated, uses molecular oxygen to oxidize this position of the E ring and release light. So if you follow the reaction using UV vis, there's chromophore changes when you go from luciferin to oxyluciferin, which is shown here. And so Stoyanovich and Kishi, this group synthesized the chromophore of luciferin, so just the D, E, and C rings of luciferin. and they observed sort of the spectral properties of this substrate mimic. And they're very, very similar to the full-length whole luciferin substrate. The excitation lambda max and also the emission lambda max are very close to one another. And also, this actually can serve as a poor substrate for the enzyme. So you can actually feed this to activated luciferase and get a faint glow. So this, I think, is a good mimic of the substrate in general. It has all the features you need to form an excited state that can emit light of close to the right wavelength. So we use this as sort of a minimized substrate in our calculations. So we wanted to sort of see how good our methodology was in reproducing sort of the spectral changes during the reaction. And the overall pattern is very similar to what is observed. You see this decrease in wavelength for luciferin and the increase for the oxyluciferin. But it's underestimated by about 50 to 70 nanometers, both the excitation wavelength and the emission wavelength calculated compared to experiment. So it's reasonable, not super accurate, but I think it gives us a good idea. It's a reasonable method to use to get some insight. So we used this time-dependent, long-range corrected density functional theory, this omega B97 XD functional, as our computational methodology. And so when this reaction starts, it's been proposed that some sort of catalytic base, now we think it's this glutamate residue, this E1105, which moves into the active site, can serve as the catalytic base to activate luciferin as an enolate. And then this enolate can react with molecular oxygen to eventually form this excited state and relax and produce light. But the structure of that excited state is unclear. So we considered several different possibilities. So you can maybe react this enolate with molecular oxygen to form hydroperoxide or peroxyanion, or maybe this gemdiol or gemdiolate intermediate. These are potential luminophores. So we'd like to calculate the ground and excited state energies of each of these, calculate the emission wavelengths for each of these, and see does any make sense? Does it match up closely with the experiment or not? And we also varied the medium. We did this in aqueous solution, pertinacious solution using a dielectric of around 4. We also considered different stereoisomers, R versus S, and then also different orientations of this oxygen bond, and screened all of these to see if we could see any trends. So the first one that we looked at is this peroxyanion intermediate, and it has a calculated emission wavelength of between, say, 550 and 600, which is a little high. Again, 475 is the experimental emission wavelength. And also, this is going to be underestimated versus reality. So it's kind of way too high to be feasible for this bioluminophore. But we noticed if you form a hydrogen bond, so some of these orientations are capable of forming sort of intramolecular hydrogen bond. So for the E isomer, this peroxyanion can form a hydrogen bond with the D ring nitrogen. So if you do that, then you shift it to lower wavelength, okay? And you also increase the intensity of the emission oscillator strength. So that kind of suggests the active site, if you form hydrogen bonds, you can sort of tune the wavelength. So that's possible. And also if it becomes protonated, then it's more in line with the correct wavelength range. And the other thing with the peroxyanion, which really rules it out, so if you form the ground state, it's slightly favorable to form the ground state peroxyanion with the E isomer, but it's not really favorable with the Z. And then if you form the excited state, it's like 60, 70 kcals higher. So it's way unaccessible. So it's not likely to be involved in this reaction. But if we look at the hydrogen peroxide, so the protonated peroxyanion, it has a wavelength, emission wavelength now closer to what we're looking at, 440 to 480. So that's not too bad. its ground state is significantly lower, minus 17 kcals per mole relative to luciferin. But again, the excited state is kind of high, 55 kcals per mole. That might not really be accessible. So we don't really think it's the hydroperoxide. But if we look at the gem diol, that one, again, is sort of between 500, 510 nanometer range. So that's a little high yet, but it's very favorable. So the ground state gem diol is almost negative 90 kcals per mole. So it's very favorable to form this gem diol. And even the excited state is favorable, but the wavelength is a little off. However, if you deprotonate the gem diol to form the gem diolate, the wavelength gets a little bit higher, but the energy also gets a little bit higher. So it's in the proper range, this gem diolate, to be the correct emission wavelength. It's favorable to form the ground state, and it's slightly disfavorable to form the excited state. So it's only plus about 7 kcals per mole. So that's reasonable to form that type of intermediate. So we have here this gem diolate is a pretty good candidate for the bioluminophore. Either a gem diolate or partially hydrogen bonded gem diol could be the bioluminophore, right? So if we actually look at the molecular orbitals of this emission process, so if we're in the ground state, this will be fully occupied, and this would be the highest unoccupied molecular orbital. But if you're forming the excited state, it's going to be an open shell singlet. So you have one unpaired electron in each of these orbitals. So you have one electron here and one electron here. And then, so it's almost two open shell singlets, so two unpaired electrons. So if you make a transition from this energy level down here with the emission of light, you take an electron out of this orbital, which is sort of centered mostly on this carbonyl, and put it into the conjugated pi system of the molecule. So if you look at that and kind of put it into a mechanism, this is kind of what you can come up with a potential mechanism. So you start with your luciferin substrate, you activate it as an enolate. This can react with molecular oxygen. Maybe you can form this sort of dioxetane ring, which is similar to what is observed in firefly bioluminescence. And then you can get an intramolecular electron transfer from this ring to induce the cleavage of the OO bond to form this open shell singlet. And then you transfer an electron from the carbonyl, as in the figure, to the conjugated pi system to release light and form this ground state gem diolate. And then you can eliminate water and form the final product. So this is sort of our working hypothesis for how this enzyme works. And this reaction is similar to, again, what is observed with fireflies. And there it was proposed to be this chemically initiated electron exchange luminescence reaction, the SEAL reaction. So it's similar to the SEAL mechanism, possibly for dinoflagellate bioluminescence. So I didn't really mention, but this is specifically for the E isomer of luciferin, and it's not really clear what the actual configuration of the substrate is. E was favored in that synthetic study because it was the more stable isomer, and its spectroscopic properties were slightly closer to the purified luciferin. But luciferin is thought to come from chlorophyll, and chlorophyll has the Z isomer. So it's possible it could be either, right? And it's not really clear. So if you look at the Z calculations, it's very similar. Its ground state is very similar to that of the E isomer. But interestingly, the first excited state twists. So the two rings become perpendicular to each other during the calculation. And there's a very low lying excited state, which is dark. So that's not likely to be the excited state relevant to the emission process. But there's a second excited state, which is pretty close to the proper emission wavelength. Okay. And the same thing with the Z isomer. Again, the energetics are close, pretty favorable to form both. And you have the second excited state, which could be in the proper range to serve as the bioluminophore. So if you look at those orbitals here, so here is the fully occupied. You have this low-lying first excited state, and then you have this higher second excited state. So this is the orbital for this one. And so if you're in the excited state, you have one unpaired electron in each of these orbitals. And then you, during the emission process, you take that electron from here, from this ring, transfer it to the other ring in this region of the molecule. So you can write that on paper as a mechanism. So again, you sort of activate the substrate starting from the Z isomer of luciferin, form an enolate, and then react with molecular oxygen to form this dioxetane ring. And then you would twist the molecule to unpair the two electrons to form this open shell singlet. And then you can maybe induce a deprotonation when you twist and open up the OO bond. And then you can do the intramolecular electron transfer from one ring to the other to form this twisted intermediate. Then you could dehydrate and twist back to form the same final product. So, so this is actually involving a twisted intramolecular charge transfer. That's actually a thing. I didn't really realize that until I got into this. So it's a very common reaction found in photochemistry, but there is no biological example of that. So if that happens to be the case, that would be pretty interesting, the first biological Tict reaction. But I think it's one possibility. The calculations suggest it could be possible. So in conclusion, in the second half, we used a variety of computational chemistry methods to look at this interesting enzyme. So we used constant pH, accelerated molecular dynamics. We got some good insight into the mechanism of this pH activation of the protein. And then we used time-dependent DFT to look at a potential luminophore. And we think most likely it's a gemdiolate or hydrogen-bonded gemdiol, which is the luminophore. And it could either go by a seal or a Tict mechanism. So future work is going to be focused on that. All right, so then just in summary, just like to thank all the people who actually do the work. I just kind of sit behind my desk and play on my computer, right? So we have 2 postdocs and 7 grad students currently. One graduate student graduated fairly recently. He did a lot, Feng No, he did a lot of the dinoflagellate luciferase work. Most of the F430 biosynthesis was done by Kaiyun Zhang. He had this science paper before he went through his qualifier, so he was a rock star, right? So he did pretty good starting out in the lab. And then, of course, the funding. Pablo mentioned NSF and DOE as the primary funding sources. So with that, take any questions. Thank you.
I'm not an expert in this particular topic, but it might be a little naive, but your molecular dynamics experiments where you predict how the active site is exposed or not in the luciferase, do you actually accompany those predictions with experimental data that actually shows that those residues are critical for the model that you proposed?
Right, that's what we're doing now. Yeah, we'd like to use this as sort of a guide and make all the mutants and apply several different techniques to show that that's the case.
One more thing.
Right.
Your glutamic 1105 residue, quite intriguing. I was wondering if you had considered to mutate that one to a lysine residue, which completely changes charges, to see whether actually you can abrogate that, flip.
Right. Yeah. So.
Suggestion that.
Yeah, definitely. So the suggestion was, repeating the question for, was mutating the proposed catalytic glutamate to a lysine and see if that abrogates activity. Definitely, we're going to try that. The charge reversal and even alanine or aspartate, make it a little bit shorter and see if it changes the activity. Definitely. Yeah.
So in your luciferase acid, I noticed that there's ATP missing in this. So is this an ATP independent assay?
Yeah, this enzyme does not use ATP, so it's different than the Firefly case. Yeah.
And so does the Firefly is able to use any of your gym dials or any of those analogs for it's essentially a chopped off version of the luciferase?
Yeah, so that's kind of the interesting thing with these luciferases. So they're independently evolved, and the enzymes are all different, and the luciferins are all different. So the structure of the Firefly luciferase is completely different than, even though it forms a dioxytenone type intermediate. The structures are completely different, so they won't cross-talk to each other.
I have one question. So how can you make sure that we cannot get the coenzyme F430 right in vivo? It's because that the step contains the CFBC and D, right?
Yeah, yeah. So the question was, how do we know that the CFBCD reaction wasn't working in vivo? So we just assume that that's the stopping point, the problem, because we see its substrate accumulate and we don't see anything after that step. So,
I have one suggestion. So because we can get the enzyme in vitro by add the enzyme step by step, right?
right.
So how about we put this reaction system into the cell lysate of E. coli?
Yeah, so if you take the cell lysate and use that, the enzyme is active, right? But we're using dithionite as an artificial reductant. So that's the trick, is how do you get E. coli to do that reduction in vivo? So we think it might be a ferredoxin problem. It doesn't have a proper ferredoxin to recognize. So we're testing that. Yeah, right.
In your calculations to get the emission maximum from those compounds,
Right.
if you apply those same methods to known fluorescent molecules, how accurately is it predicting the emission maximum?
Yeah, so the question was, how accurate is the method in calculating these things? And that's why we did it first, the calculations first on the luciferin and oxyluciferin, the excitation maxima and the emission maxima, and compared it to the known literature for that. And it's about 50 to 70 underestimated in both cases.
That's why you're using underestimated.
Yeah, right. So, yep.
Well, before we let you go, Steve, I'm going to give you this little bag with some souvenirs.
All right.
You know,
Thank you.
T-shirt, a fun T-shirt. And
Wow.
you know, mug. Lisa,
Fancy.
Thanks, Steve, for a great seminar.
Thank you.
So next is lunch with some more practices. Great job. So remind me tomorrow morning to get the.
No, he said
You
I
don't
can
need
take
it
it now.
Okay.
That's what he said. He's not going to work.
Nice talk. See you at 4. Very nice. Yeah, really. Look forward to seeing you later.
Hi, I don't get to see you later, but I have a couple questions.
Sure, yeah.
What program did you use for your molecular dynamics calculations?
So we used Amber.
Amber? Okay. And what cloning method did you use for the project in the first half of your talk? Because you did 5 in like a year or two. That's
Right. Yeah, I mean, so we just did, we ordered the genomic DNA, and we just did PCR to clone each one. And then, so we did a combination of either restriction cloning or Gibson assembly.
okay.
It's just because
All right.
here in the
Thank you.
Hi, my name is Catherine Creed. I just had 2 quick questions about your presentation. So for the TICT synthesis, you were talking about how like there was like a step, it was before the UV light, you had a double bond and you said you twisted it and it like divided up into 2